Hello everyone. Today we are completing our leftover portions. I guess leftover portions about human origins, like the Piltdown Man. Today there will be some mispronounced words or errors. So sorry for that. So let's start learning. Like Piltdown Man. This relates the story of how a fossil discovered in 1912 in England influenced our thinking about the origin of humans until it was revealed that the fossil known as Piltdown Man was a clear fake, clever fake, a combination of human skull and the lower jaw of a female orangutan. By about 1915, most anthropologists had agreed that early humans had large brain cases, ape like jaws, and large canine teeth. Evidence for this interpretation came from fossil rhinium and lower jaw found in 1912 at Piltdown, England, by an amateur British archaeologist, Charles Dawson, in 1864 to 1916, and named Eoanthropus dawsoni. Some pictures might be scary for someone, so sorry for that. This is just an evolution class, human evolution class, nothing else. You don't have to be scared of this. So many anthropologists accepted the Piltdown fossil as valid, a situation that continued for about 40 years until it was shown that the entire fossil was a hoax. The teeth had been artificially grounded down. The rhenium was of a different age than the jaw. Artificial pigmentation had been used to color the bones, and the molar teeth had long roots like those of apes. Moreover, the associated animal fossils at the Piltdown site had a large accumulation of radioactive salts whose origin could be traced to a site on Tunisia. So, Piltdown Man turned out to be a fabrication, a human rhenium and the lower jaw of a female orangutan, a hoax perpetrated by someone who knew enough to destroy all obvious signs of the specimen's true origins by removing the jaw joint and modifying other key features. In a book on the specimen published in 1990, Frank Spencer argued that the Piltdown forgery was the work of its principal discoverer, Charles Dawson, in a conspiracy with Arthur Keith, a leading Scottish anatomist and physical anthropologist. The historians of science suggest they are suspects. Moreover, whoever perpetrated this hoax, the result was the preservation of false views with false facts, at least for a time. The events that followed showed that false facts can be challenged in science and false views replaced. Discovery, description, and naming of Australopithecus africanus laid the basis for Africa as the geographical location of the origin of humans and the place from which humans radiated to populate the entire globe or out of Africa hypothesis. Then dwarf hominins on the islands of Flores. Further to the discussion of human origins, more information is provided about the discovery of fossils of population of pygmy hominins who lived as recently as 18,000 years ago on the island of Flores in Indonesia. The cause of enormous excitement and controversy, this population has variously been interpreted as a relicit population of Homo erectus, a new species of humans like Homo floresiens, or a dwarf and congenitally deformed population of Homo sapiens. These widely divergent conclusions are based primarily on analysis of one complete skull, analysis of long bone sizes and shapes, and the presence of tools normally assigned to more than one species of Homo. Then discovery of what has been interpreted as a dwarf, 1 meter about 3.3 feet tall, or pygmy population of Homo erectus on the island of Flores in Indonesia is so recent and the findings so unexpected that analysis and interpretation is ongoing at a fast pace. So especially controversial is whether these individuals represent a new species of Homo Floresians as a illicit population of Homo erectus, or a dwarf or isolated population of our own species Homo sapiens. Assignment to Homo erectus would mean that a remnant of this species existed 18,000 years ago. Specimens from the site date back to 95,000 years ago. The presence of tools more advanced than those of Homo erectus is known to have used is evidence against assignment to Homo erectus. Analysis of 140 cranial features led investigators to conclude that these individuals represent an early pygmy population of Homo sapiens with signs in one complete skull. An adult female unearthed at Luang Bua of a congenital malformation similar to a malformation called microcephaly, known to occur in modern day humans. So, microcephaly is a condition in which individuals have small brains and therefore small skulls, which enclosed a brain of 400 cm cube is comparable in volume to the brain of a chimpanzee and compares in volume and other measures with microcephalic human skull. So, in this picture, you could see the skull of the most complete hominin from Flores dates at 18,000 years before present, like shown in the left, compared with the human skull on the right. This individual is reconstructed as having stood a little over 1 meter in height, with long arms in relation to the length of the legs, unusual shoulders, and a chinless mandible. So much of the analysis has been conducted on this one fairly complete skull, which is shown in the figure. Material from a dozen other individuals remains to be examined, so caution is required when drawing interpretation or conclusions. So later enlargement of brain size during primate, including human evolution, has been associated with enhanced function of MCPH genes, which regulate the development of nerve cells in cerebral cortex. To explain the diminutive size of Flores individuals, Richard in 2006 invoked mutation in the gene of this microcephalin MCPH1, one of half a dozen genes in which mutation results in microcephaly. Another study amassed evidence that a mutation in the gene for the growth hormone receptor made Flores individuals insensitive to growth hormone. So, disruptions of growth hormone would upset the growth hormone insulin like growth factor 1 axis required to regulate growth. Consistent with this hypothesis, individual extant humans with the inherited disorder Laren syndrome, Laren type dwarfism, has a mutation in the growth hormone receptor, making them insensitive to growth hormone, resulting in short stature. So, cretinism also has been invoked to explain the small brain volume and features of the Flores skull. Oxnard et al. In 2008 provided evidence for myxoedematous endemic cretinism resulting from congenital hypothyroidism. Both brain and skeletal size are reduced in individual modern humans with cretinism as a sequence of underactivity of the thyroid gland. Local high incidences of cretinism on Flores have been associated with iodine deficiency, which remains a problem for individuals living on Flores today. So, by definition, individuals with microcephaly have small skulls in relation to body size. More recent analysis of bones from 7 Flores individuals led to the conclusion that the size of the skull is proportional to the size of postcranial skeletal elements, a finding that is inconsistent with microcephaly but not with dwarfism. Extinct hippopotamus from Madagascar, which have 30% smaller brains than predicted from hippos on the mainland scales to the same size, were used by Weston and Lister 2009 to demonstrate that this Homo floresiens could have evolved its relatively smaller brain in response to isolation. Additional factors involved in the evolution of the human brain are outlined elsewhere in this text. So, interpretation that Flores population is an inland island adapted population of Homo sapiens, hence the small size, perhaps with some individuals expressing congenital abnormalities, was reinforced by the discovery of March 2008 of the remains of a population of small bodied humans who lived in caves in the western Caroline island of Micronesia. Then, on the other hand, analysis of the feet of Flores individuals led some investigators to conclude that the feet are those of a basal biped, comparable to those known from 2 to 3 May by old human ancestors. The feet of that Junkers and colleagues classify as Homo floresiens are exceptionally long for a species in the genus Homo, a feature interpreted by Junkers et al. In 2009 as basal for hominins, calling into question the origin of this Homo floresiens from Homo erectus. The large hairy feet of hobbits in J.R.R. Tolkien's epic trilogy of Lord of the Rings are invoked in comparison. With such divergent structures, mosaic evolution, pathology, the jury remains deadlocked on the precise identification of this population. So, this remains as a mystery. That is all I wanted to say. Stages in the manufacture and use of tools by species of Homo. As introduced in the accompanying chapter, tools used by species in the genus Homo dating back to 2005 2.5 MYA are assigned to 4 stages. Characteristics of tools from these 4 stages and their assignment to particular species of Homo are outlined. Tool making and tool use is classified into 4 stages and related to species of Homo that made and used each tool type, beginning 2.5 MYA and with Homo habilis and extending to Homo sapiens. Soldovan tools consist of sharp-edged stones that could be made with a single blow of one rock against another. Such tools are sufficient to strip tough fibers from plants, break up animal carcasses, and expose the narrow cavities of long bones. Soldovan tools date back to deposits that are at least 2.5 MY old and are found in deposits over the ensuing millennia. Some paleoanthropologists suggest that although Moldovan tools are traditionally ascribed to Homo habilis, they may have been used by earlier hominins. Then, Herculean tools consist of stones tipped from both sides using multiple strikes to create the cutting edge. These tools are known as hand axes over 500,000 years ago and were in use up until 200,000 years ago. And axes are most abundantly found 1.5 MYA associated with Homo ergaster and Homo erectus. Then Mousterian tools require some complicated working and reworking manufacturing than the other types of tools. These Mousterian tools first appear 200,000 years ago and are found in 40,000 years old deposits. They are associated with Homo neanderthalensis in Europe and with both Neanderthalensis and Homo sapiens elsewhere. The presence of points indicate tools made for hunting, with the implication that the points were affixed to long sticks or spears. The presence of scrappers indicates tools made for scraping animal carcasses, either for food or for skins to be made into clothing. So, this picture shows early Stone Age, Lower Paleolithic Moldovan tools, then Acheulean tools, then Mousterian tools, Middle Stone Age, Middle Paleolithic tools. So, stages in tool development in the Paleolithic Stone Age begin with choppers and scrapers of the Moldovan stone industry. Then, Acheulean tools consisting of clevers left and hand axes right side, and most abundant 1.5 MIA and used by Homo erectus and Homo ergaster. Then upper Paleolithic tools used by Homo sapiens include hooks used for fishing and needles for sewing. These were the dominant tools in Asia and Africa between 90,000 and 12,000 years ago. Specialists divide the Upper Paleolithic tool period into temporal and species specific subperiods, reflecting increasing sophistication and diversified use of tools and other objects. So, ivory beads and carved female Venus figurines are first found between 28,000 and 22,000 years ago, depending on geographical location. So, barbed harpoons, spear throwers, and the first rock paintings appear 18,000 to 12,000 years ago. Following this period are the Mesolithic and Neolithic ages, the later beginning about 10,000 years ago and marked by polished stone tools, pottery, domesticated animals, cultivated plants, and woven cloth. Then next topic, speech and language. The anatomical and genetic evidence for why humans possess speech and language, why chimpanzees do not, and whether Neanderthals were capable of speech are discussed. The most symbolic of human vocalization or animal vocalization is human speech and language. The sound producing systems of modern humans and our closest sister group, the chimpanzees, have been investigated to determine why humans have speech and language but chimpanzees do not. In the human sound producing system, the larynx, just look at the figure, acts like a reed in a woodwind instrument. Sound is turned into the basic units of speech like vowels in the pharynx. The length and shape of the pharynx determines the frequency pattern of the sound emitted and so modulates vowel sounds. As the size of lower jaws are reduced during human evolution, the larynx moved to a lower position and the tongue formed the anterior wall of the pharynx. So the chimpanzee tongue is isolated from the pharynx. Just look at the figure. So that air is expired through the nostrils rather than through the mouth. When combined with the inability of chimpanzees to manipulate the tongue within the pharynx, the anatomical limitations to speech in chimpanzees become evident, although numerous attempts to teach chimpanzees to speak have been made. So when we just look at the picture, upper respiratory systems of humans, chimpanzees, and Australopithecines showing structures associated with vocalization is shown. The pharynx is much longer in human adults than in chimpanzees, enabling humans to enunciate vowels and symbols by positioning the tongue in both mouth and pharynx. So newborn human infants show the same overlap between epiglottis and soft palate as a non-human primate. But the pharynx lengthens considerably during infancy and childhood. A reconstruction of the presumed upper respiratory system of an adult Australopithecines is shown in the D picture. As in non-human primates, the epiglottis overlaps the soft palate. The back of the tongue does not reach the pharynx, and the larynx is relatively high in the vocal tract. Chimpanzees do communicate extremely effectively with facial expressions, body language, sounds, both among themselves and with human trainers under controlled conditions. So, the position of the larynx and the size of tongue in relation to the length of pharynx in Australopithecines show relationships similar to those seen in non human primates, supporting the hypothesis that speech arose only in later lineages. Neanderthals had a chimpanzee like vocal tract. It has therefore been concluded that Neanderthals were not one of the lineages in which speech arose. However, the acquisition of DNA sequence from Neanderthals is consistent with different interpretation. The gene forkhead boxP2, like FOXP2, has been linked to speech in humans. So FOXP2 is found in most mammals, but the human gene contains two mutations not found in other mammals. Nucleotide substitutions at positions 911 threonine replaces aspartic acid and 977 like arginine replaces serine of exon 7. Humans with a single copy of the gene have speech impediments, and mice with a single copy have much reduced vocalization. So, Neanderthal DNA contains the same two mutations found in humans. Chimpanzee lack the mutations Clearly, it is tempting to link these two mutations either to the acquisition of a speech vocalization or one or more important aspect of speech, and to include that Neanderthals had some form of speech and that chimpanzees lack speech because they lack these mutations. So, diagrammatic views of how adult humans produce three vowel sounds by positioning the tongue, which is represented with the help of arrows. It is represented with the help of arrows in different parts of the oral airway. Note that a sharp bend formed by the hard palate above the mouth and rear wall of the pharynx partitions this airway into right angled mouth and pharyngeal sections that are essential for this vowel sounds. So then we learned about that gene. Various aspects of speech and language have been traced to particular areas of the brain. On the temporal lobe of the left cerebral hemisphere is Wernicke's area concerned with formulating and comprehending intelligent speech. Individuals who have lesions in this area emit bodily babble that conveys no information. Individuals with lesions in Broca's area on the left frontal lobe, which coordinate vocal muscular movements, have difficulty speaking. So Broca's area lesion lead to difficulty speaking. Then aspects of language such as grammatical structure and vocabulary seem associated with neural circuits in the prefrontal cerebral cortex that lie somewhat forward to Broca's area. This proportionate enlargement of the prefrontal cortex is an obvious feature of the brain of Homo sapiens. So it has been hypothesized that tool use and oral language share a common neurological evolutionary basis. Both language and manual tool manipulation are sequential processes. Appearance of stone tools coincide with the appearance of Broca's area in hominids is consistent with the functional connection, through it does not prove such a connection. So linkage between the two functions is consistent with their common localization in the left or dominant hemisphere of right-handed individuals. For those left-handed people whose manual control center lies in the right hemisphere, language control is often found there as well. So in this picture we could see Wernicke's area in the left temporal lobe and Broca's area in the left frontal lobe of the human brain, and this both sides associated with speech. So frontal lobe is there, motor cortex, Broca's area, olfactory bulb, primary auditory area, temporal lobe, pons, medulla oblongata, cerebellum, occipital lobe, primary visual cortex, lunate, sulcus, angular gyrus, Wernicke's area, parietal lobe, somatic sensory cortex, then central fissure. These regions are also shown.